Welcome to Genetics in Pills. So, in the previous pills, we saw how it was possible to demonstrate that the DNA is the key molecule for the storing the genetic information of organisms. And we also saw the structures of the nucleotides, the basic molecules composing the DNA. Here you see what we showed last time. You can see the purines with the 2 rings and the pyrimidines with the 1 ring. So we have adenine composing the deoxyadenosine, mono, D or triphosphate, the guanine composing the deoxyguanosine, mono, D or triphosphate. We have the cytosine composing the deoxycytidine, mono, D or triphosphates. And we have also the thymine composing the deoxythymidine, mono, D or triphosphate. Now, let's discuss the Chargaff rule, studied about in 1949. So Chargaff digested the DNA and analyzed the composition in basis of the DNA from several organisms, and found either in Mycobacterium tuberculosis, in yeast, in various other organisms from the sea, like this one, Paracentrus lividus, and in herring, in rats, in Homo sapiens have different tissues like thymus, liver, spermatozoa. In other words, in all the tissue and organism analyzed, he found that the molar content, so the number of molecules corresponding to adenine, was always similar in the range of the experimental error with the number of thymines, so the molar amount of thymines, and the number of guanine was similar or identical to the one of cytosine. This is the molar percentage, and you can see that all the time there is a correspondence between adenine and thymine and guanine and cytosine. So an important rule was finding that over the time we have some amount of adenine, we have the same amount of thymine, and we have some guanosine or guanine, and we have the same amount of cytosine. So what was this strange rule? The other question was, is it possible that different organisms have the same composition? And the answer was not. As you can see here, actually the composition in these bases was different. For example, in Mycobacterium tuberculosis, we have 15% in AAT and 35% in GNC. Rather, in, for example, human, we have about 30% in ANT and about 20% in GNC. So, different organisms, different composition, but all the time, A corresponding to T and G corresponding to C. Again, this is another the picture of the Chargaff rule. As you can see, now we know that the DNA is a double helix, but actually at the time they didn't know it. So Chargaff contributed in proposing a model, a key model for Watson and Crick in the double helix theory, was that actually each base corresponds to a correspondent different base. So A corresponds to T and C corresponds to G. But at that time, it was not possible to have this sort of model. The model arrived 3 or 4 years later. So the composition in AT, and the CG it depends on the organism, and the correspondence between purine and pyrimidines is 1:1, adenine to thymine is 1:1, and cytosine to guanine is 1:1. Thanks for watching Genetics in Pills.